Today I'm very happy to be here and share with you this data that we collected on our project funded by the Human Brain Project, that is MAPS, Mapping Brain Circuit Spatial Navigation. And this is actually a collaboration that we had, so MELE, that is my lab, with Michele Migliore in the National Research Council. We are in the Sapienza, University of Rome. And basically when we first approached the HBP community, our idea was trying to develop a network model to understand brain dynamics during spatial learning specifically. Why spatial learning? Because we have a strong background, expertise, and actually interest in understanding how does it work, spatial memory, spatial learning, and circuit underlying memory formation. And moreover, recently, actually, our attention was pretty much caught by a phenomenon that is very general, and you probably have heard about it because it's true also for human, that is known as the spacing effect. So the spacing effect is very general, as I said, and it refers mostly to the findings that spaced repetition are better remembered than massed repetition. And so in a learning task, longer intertrial interval lead to a more durable memory than shorter intertrial interval. And as I said, this is true for human, and this also recent work have been demonstrated also in animal models in a variety of learning tasks. And today I will just show you some briefly example of human study. For example, this is just a comparison between massed and distributed practice in a basic math fluency. And the point that I want to actually highlight is the maintenance that is stronger. So they have a stronger memory when the practice is distributed by time instead of one is massed or less distributed. And it has been demonstrated that this effect is due to an enhancement of synaptic plasticity. And this enhancement could actually work. There are two hypotheses at the basis of this enhancement. The first hypothesis that is most leaded to the idea that spacing effect, so spacing stimuli, could overcome refractory period of the refractory period, and so it's able to enlarge and strengthen the target. So it's working on the same target. The other hypothesis is the idea that instead, with the space stimuli, so in a spacing training, there are new spines are involved, and so there is a priming phenomenon, and so there is an involvement of new spines that then will be enlarged and strengthened in the subsequent stimuli. However, this phenomenon is still poorly understood, and is poorly understood on a cellular and molecular mechanism and on a circuit mechanism. So on a cellular and molecular mechanism, there are data, for example, from Smolen group that they demonstrated that probably the molecular mechanism behind this effect is that the space stimuli allow more than one round of transcriptional factors, so more than one cascade is activated because space stimuli are somehow longer in time, and so allow more than one cascade of transcription, and the synchronization of these basically signals allow to an increase of the signal at the end. In a more circuit view, instead, if you work with this is an example of human again, and they saw that the theta frequency in the occipital region of the brain, in posterior part, are enhanced in distributed compared to masked, in a distributed compared to a masked condition. In mouse, instead, we actually are studying the circuit behind this phenomenon. We are trying to study the circuit behind, and we just published this paper in which we found that we basically set up the masked and distributed training in mice. And we proved that actually distributed training, learning, lead to a more durable memory compared to masked. And we are looking at the brain region involved, so the circuit and the cell organization of the brain region. In this scenario today, I will talk about one specific region and the cellular organization of this region, that is the hippocampus. So we actually investigate the cellular activation pattern after a masked and the distributed training, and we evaluated how the hippocampus actually is active in those two different training protocols. I don't think there is need to tell you that hippocampus is pretty known region in spatial memory, and those are just few of the paper, but literature is full of the idea that hippocampus is needed for spatial memory, for memory formation. And today, especially, we investigated the activation of one of functional sub-region of the hippocampus, that is the CA1 region. So how we approach this study? We are more in a lab that has a strong expertise in basic science, in vivo study, manipulation, and the Migliore lab instead is more on a computational expertise and computational and data analysis. So we put together this approach, and on our side, we decided to build this, to set up this training in the mice, using this well-known task. I don't know if you are familiar, but very briefly, this was developed in 1980 by Morris, and it's basically a pool in which you put the mice inside, and the mice need to find the platform underneath the water. How we can find it? He needs to use visual cue that surround the pool, and so you need to build a spatial map to find the platform. But the first day, we just put the mice in the pool, and they just need to understand that the place is safe. The platform, in fact, indeed is visible. So they just need to understand, okay, there is an escape. Day after, we train the mice, and we train the mice into different protocol. The distributed protocol, in which basically they are trained for 6 sessions across 3 days, and each session is 2 sessions per day, spaced 4 hours apart. And in a mass training instead, the 6 sessions are given to the mice across 1 day, and they are spaced 10 minutes apart. This, I think, it's a nice video just to give you the idea. This is a rodent that just swims in the pool. This is a platform, and he's using visual cue that you cannot actually see, but he's using visual cue to finally find the platform. And then find the platform. So when we analyze the data of this behavior, and that's I show you today, we actually, this is a reminder of the protocol, spaced and masked, and this is an example of the tracking of the mouse. So when you put the mouse in the pool, usually at the beginning of the training, they don't know where the platform is, they just swim around, they avoid the center because they are scared of open space. And instead, when they, at the end, they know where the platform is, so they go directly there. And when you measure, actually, you can evaluate the learning curve, and usually the distance that they travel, that as you can see here, clearly reduces by the time, so by the end of the training. And at the same way, the escape latency reduces, meaning that the time that the mice took to reach the platform clearly reduces because they know where the platform is by the end. And those measurements gave us the idea that basically the training protocol did not affect the ability of learning. So they are learning how to find the platform, and they are learning in the same way. Not only this, but we took a measurement to see if actually the cognitive strategy of these mice was changing. And we decided to take the Tigmotaxis, that is a non-spatial navigational strategy. As you can see, the mice, this is an example, so mice swimming around the periphery of the pool. And again, this strategy usually, it's been demonstrated that decreased by the time, because by the time that they know where the platform is, they just switch to a direct spatial strategy. And so this measurement actually allow us, provide us the information that the mice are learning, and they're learning in the same way, using the same using the same cognitive strategy to reach the platform. Once we confirm this data, we collect all the brain, and we went to analyze the activation of the hippocampus and the CA1 specifically. How we decided to do that using a marker that is pretty commonly used. That is an immediate early genes. That is a FOS mapping of the CA1. So FOS basically is a marker that is used to quantify and evaluate a brain activational state. Those are just example slides of anterior-posterior different coordinates of the hippocampus that we quantify. And this is an example of how you visualize FOS. So these black dots that are the FOS-positive cells spread around the CA1. And FOS is not only very useful marker for activational state, but also correlate pretty well, and this is just an example I reported from literature, that it correlates very well with learning performances. And so it's very useful also to study specifically the process of learning and memory. So for this experiment, we got 3 groups of animals. and so many animals. We got 3 groups, masked and distributed, we are already familiar with that. Then we got the control mice, because this group was relevant to check the FOS expression baseline. So this group was just handled by the experimenter, bringing to the experimental room. They stay there, and they in the holding cages, and then they, at the end of the training, they came back in the facility, in their home cages. The point that I want to stress is the fact that this is the first work that it's really give a detailed map of the FOS and the activation of CA1. We really collect a big amount of data, and that's the strength of our collaboration, because we could join the other lab, Migliore Lab, and analyze all this data. Because basically we provided a map of FOS of all, from the light anterior, the light green, the anterior slice of the hippocampus, to the posterior, there are the dark green, for roughly 12 or 14 slices per animal. And then we also detailed the septotemporal axis of the CA1. So we decided one reference point for all the animal. We designed 12 regions of interest. There are from our normal for like ROI, just to be shorter. And those ROI are just provide a detailed information on how FOS is expressing along all the mediotemporal axis. And as you can see in the very anterior slice, there are no temporal ROI, because the CA1 is very short and it only fits 7 ROI. First things that we did with this data, we tried to see how the expression of FOS change in the group. And so, okay, it's not the great view, but anyway. So in control mice, the expression is really low, and that's somehow expected, because they don't do any learning task. They just bring it there, and they are in the holding cages. In masked animal, instead, there is a peak of activation, as you can see here, in the temporal central area of the CA1. And very nicely, in the distributed, there is a shift of activation. Indeed, in distributed mice, the activation is higher, there is an increase of activation, and it's localized in the posterior area of the CA1, actually. And when we decided to quantify this, we grouped the ROI, so the signal per ROI, we grouped by 4. And we obtained actually a significant difference in distributed mice along all the septotemporal axis. We actually didn't reach a significant value, but clearly there is an increase in the mass expression here in the temporal area. Said that, we wanted to go further in detail to see if this pattern could be really strong and really consistent. Because as you know, the mice, like the individual variability is something that I also heard before, is something that like affect the experiment, and it's important to check. And actually the data that I show you left us very enthusiastic, because first of all, we decided to analyze the data choosing a threshold. So we analyzed the frequency of the region of interest within certain cell. And we evaluated in the control there are more region of interest with less cell. Expected, because probably the activation is low. This is not true for the distributed, in which there is an increase between 30-40 cells. At this point, we cut off at 23 cells, and we decide to see what happened above this threshold to try to avoid the specific activation of FOS that could be due to the fact that the mice anyway is grooming, is sniffing, is looking around, and somehow FOS is active. And so we analyze the data again, and those are all the animals, so each square is one animal. And basically, the control have the low activation above threshold, and those are this light green square that you can see highlighted here. And this is actually 3D reproduction that we obtained using the Queen software that actually we came to know thanks to another meeting that we had in Athens in 2020. And so this is a very nice software to analyze all of this data. And when we look at the mass instead, the activation is higher, so confirming what we already seen before. But there is something that very strong. So there is a consistent subregion inside the CA1 that is active in all the mice, regardless of their variability. So there is something that is true for all of them. And the same outcome we found in distributed mice, with the difference that distributed mice have a larger activation. The area is larger and is posterior here, as we found already. And this is possibly could explain actually why the memory is stronger, probably because the activation of the region is larger. And is consistent again, is the same region in all the mice. So that's actually pushes us to ask and to ask if we could use this information, this pattern information, to discriminate the three groups. And so we train learning algorithm, and there's a part that actually was done by a postdoc in Migliore Lab. She basically trained, so we put all the raw data about false expression. We use different learning algorithms. We process the data, and we went to look if we were actually, we were able to find three groups. So we were able to discriminate the three groups. And very nicely, with some of the learning algorithms she tried, the accuracy of discrimination was 100%. So basically, the system was able to discriminate if an animal was trained with a mast distributed or was just a control mice, meaning that the area that was active was so specific and so relevant for the spatial information that was able to discriminate by the other training protocol. Indeed, this is a work that is almost done, fortunately, so we are concluding that. And so it's very nicely that we identify CA1 subregion for the first time, CA1 subregion that are differentially active in two different training procedures, that are consistently active in all the mice we train. And in the distributed, actually, these regions are even larger than mast mice. And so that explains for us the superiority of distributed training over a mast. Clearly, we need to demonstrate again this, but there could be a suggestion. And again, this pattern, activation pattern, was so unique that clearly can be actually be discriminated between the three groups, the two groups, using the learning algorithm. The last part of actually our paper is focusing on the clusterization of the cells. So when I show you this graph, I told you that we use it to cut off a threshold, and that's true. But we also find actually that maybe, so it let us think that maybe this cell, the false positive cell, are actually packed in a different way in the trick condition, because there is actually an increase, there is a shift in this curve. So it could be for us that the cells are differently packed in the trick condition. So maybe the clusterization level is different in mast over training, over distributed training mice. And we actually have data about the single cell position. So we have position of all the single cell in all the slices for all the mice. So we are now doing this work, evaluating how the distance between the cell, among the cell, change in the trick condition. And so to see if it actually increases, decreases, and so there is a different level of clusterization. So that's actually very like for me, of course, that's very nice work. And so, but that was a very interesting finding. And so we actually love, for future perspective, we would love to manipulate exactly this region. So we are, as I said, we are a lab that works with basic science, in vivo surgery, in vivo manipulation. So we were thinking to go directly in vivo to this region. We probably need to clearly characterize the cell, otherwise we are not targeting the cell exactly. But we would like to target the exact region that is active in mast over distributed training mice, and to see what happened. And then in a big picture, as I started the talk talking about a network models, and we are working on that too, and we are actually quantifying. Okay, we are actually, we are actually quantifying the FOS through the whole brain. We are doing this already, and we collect many data after spatial training. So we have, again, these three groups. We have the whole brain of many animals, like 10 to 12 per groups at least, and we are quantifying all the FOS data inside using the software I mentioned, the Queen software. It's very smooth, actually, and we are building correlational graph, because the idea is actually understanding how the brain region involved differently in this training are communicating together, and if there are some hub region that we can somehow target to eventually maybe an outcome of. We are not a preclinical lab, we are not translational lab, but probably this outcome could be very useful to study, because memory, you know, like there are many diseases that are like correlates, in which there is actually like a degeneration of memory. And so I really want to thank you, the people that work with me, and my PI, of course, and all the people that work with me to reach this goal. And thank you.